am Célia Miguel. I am a professor at the University of Lisbon, Faculty of Sciences, and I collaborate within the BioData project with ITKB and IPET.
One of the key resources that we have provided, the Core Code Genome Portal, but also on standards and resources required for making plant data findable, accessible, interoperable, and reusable. So for those of you that are not familiar with Elixir, it is the European infrastructure for biological data. It was founded by the end of 2013. It is an intergovernmental organization with currently 22 member countries and 1 observer, and it brings together resources in the life sciences across Europe. And these resources include databases, software, training materials, standards, cloud storage, supercomputers, in order to researchers find it easier to access their data, to analyze data and share data, but also to exchange expertise and implement best practices when it comes to scientific data. So the Elixir activities are run by 5 platforms that provide services in particular areas. And we have, for instance, the compute platform, the data, the interoperability tools and training platforms. And these platforms are driven by a number of Elixir communities that gather scientists in a specific domain, like for instance the plant sciences, the metabolomics, the proteomics, the marine metagenomics, the microbial biotechnology communities, among others. So these scientists develop standards and services and also training materials that are targeted for their own research domains by interacting with the 5 platforms. So the Elixir members work through a hub and nodes model, and this means that the hub is like the headquarters that coordinates the work across all Elixir nodes. And this hub is based at the Wellcome Genome Campus near Cambridge in the UK. Each member state establishes its own node, which is a network of the organizations in that member state, which usually includes centers of excellence in bioinformatics. And currently, there are more than 700 researchers from 220 research institutes all across Europe that are involved in Elixir. So the BioData, which is the name, as I mentioned, of the Elixir node in Portugal, is currently a consortium of 12 partner institutions that are spread across the country and include universities, research institutes, and a biotechnology industry organization, which is there's Portuguese companies in biotechnology and life sciences sector, but there are at the moment other institutions that are in the process of joining BioData. So this is the general organization of the Portuguese node. It has a management board in which the head of each partner institution is represented. We have also a scientific advisory board with members from the industry and from academia, and we have a board of directors, but the operational infrastructure is organized like Elixir in communities that focus on specific life science domains, and we have here the agro-food and forestry today, but we have other communities that you'll have the opportunity to hear about, like the sea and health domains. And then we have the platforms that are transversal to all these communities and develop the activities, for instance, in computing and training. And then we have the services that are provided by the BioData to the national and international community. So the plant institutions that are represented in BioData are the IBET, which I am representing here, the ITQB, IGC, all located in Oeiras. Then we have CEBAL in Beja and INESC-ID. And INESC-ID is not conducting research in the plant domain, but it has been actively working and committed to the development of the resources in the plant sciences. So they are also acting at the platform level. And this community became active during the implementation of the Elixir. We were participants in this H2020 Accelerate project, and we were co-leaders of the plant sciences use case in one of the work packages, with the goal of supporting the publication and access to phenotypic and genotypic data, annotated according to the standards that have been established or recommended by the community. And initially in BioData, one of our target use cases was in the woody plant domain, in part because many of these species are very important to the economy of the country. And of course, the objectives are also extensive to other plant species and contribute to address some of the challenges that we face today at the global scale, which include the sustainable supply of food and non-food materials, a competitive life sciences sector, and environmental protection. These challenges can only be addressed by significant advances that can be obtained in plant breeding from the integration of genomic or genotyping data with phenotyping data that can be from different types, like field and greenhouse experimental data, molecular data, omics, and image data, among others. So a systematic study of phenotypes on a genome-wide scale that are obtained under different environmental conditions and their association with genomic data are being widely adopted by plant breeding programs from different plant species, from traditional crops to trees. And this is reflected in the exponential growth of data in these areas in the last few years. So data sets are growing both in number and in size, but there are still a number of gaps that make these genotype-phenotype associations very difficult to approach at this moment. And some of these gaps are that plant data are very heterogeneous and multidimensional. They are obtained from different plant species under different settings and designs and conditions. They are dispersed, and unlike other publicly accessible and curated archives, for instance, for DNA and protein data, there has been no equivalent stored in multiple systems. And even when we can find these data, they are often of poor value because they are not standardized and they lack a good annotation. So the major question here that we are trying to address is how to make plant data findable, accessible, interoperable, and reusable, in other words, compliant with the FAIR data principles, which are very important to enable further knowledge discovery. So in order to help addressing this question, we have developed a number of activities in the plant sciences community, many of the times in collaboration with other Elixir nodes that are involved in the plant sciences. And our objectives have been directed towards the development or recommendation of standards and ontologies that allow plant data to be fair, in the development of repositories for plant phenotyping and genomic data, as we will describe, as well as in the development of user-friendly interfaces that allow for data deposition and retrieval. We have also been involved in the annotation and the provision of curated plant data sets and of plant data analysis tools. But of course, to ensure that these resources are adopted by the potential users in the plant community, we have also invested in the provision of training materials and organization of training sessions, and engaged also with the industry through collaborative projects to exchange and apply this knowledge. So a number of species have been prioritized in BioData Plant Sciences Community, of which cork oak is a good example, mainly because cork oak is the national tree in Portugal. Over 1/3 of the total cork oak forest in the world is located in Portugal. And in addition, we are the leaders in cork production worldwide. But of course, other species are also within our range of interests. For instance, maritime pine and eucalyptus, which together with cork oak are the most representative species of the Portuguese forest and provide important income for the country. But it is also worth mentioning other species like olive tree and grapevine, and also non-woody species like rice. So now my colleague Pedro Barros will present you in some more detail one of the key resources that has been developed by the Plant Sciences Community of BioData, which is the cork oak genome portal, that showcases several of the activities that I have just mentioned. So, Pedro.
Thank you, Celia. Good afternoon, everyone. Okay, I hope you are seeing the screen. So my name is Pedro Barros. I'm a PhD researcher at IDQB. I'm also a plant biologist, and together with Daniel Faria and an enthusiastic small team of researchers, we were involved in the development of the cork oak genome portal. So starting with a brief context, actually this new version of the cork oak DB, the cork oak database, is actually an updated version of a previous cork oak DB that was released and hosted the cork oak transcriptome database. This database was originally launched in 2014 by a national consortium which included ITQB, IBET, IGC, and CEBAL. And this consortium aimed to generate the first online database with genetic information for specifically dedicated to cork oak. And in this case, this database was obtained by cDNA sequencing in something that was originally called express sequence tags. Obviously, that at this time there was no information about the genome structure because the genome had not been sequenced yet, and it was not available. And in addition, due to the details related to the techniques used to generate this sequencing, some of the transcripts, some of the genes information were incomplete and sometimes redundant. In 2018, there was the public release of the reference genome for cork oak, and obviously this paved the way for an improved knowledge of the genome structure regarding the order of the genes, the orientation of the genes, and also the structure of these genes regarding introns and exons. And obviously during all this time, with the improvement of next generation sequencing technologies, new transcriptomic datasets, new RNA-seq datasets also became publicly available. So this was the perfect time to start updating this previous database, and from 2018 to the current year, to 2020, we worked on the improvement of this database in order to integrate the information on the genome. The goal of this QuarkOak Genome Portal is thus to create an integrated repository dedicated to QuarkOak omics. And with this, we want to develop tools for data visualization and retrieval for core genomics analysis, which can be useful for researchers in the field. And in the future, we obviously want to become a reference hub for research, which will aggregate most of the available data on genomic at the genomic and transcriptomic level, not only for QuarkOak, but eventually for closely related species, and eventually also integrate other types of data, including phenotypic data and metabolomic data, etc. Presently, the QuarkOak DB includes information on the genome sequence and also structural annotation, so we can search for genes and also coding sequences. There is also information about gene expression, so we can take some information regarding whether a gene is active or not in a specific tissue or condition in QuarkOak. And we also included a level of curation to the metadata that we added, and this is mostly related to the available studies that were present, particularly on NCBI. We improved this metadata in order to improve the clarity and precision. This portal was built using the Triple framework, which is a toolkit for construction of online community portals hosting biological data. This is an effort that is being made by an international team, and this type of framework is very important because it facilitates the integration of different types of biological data and also facilitates the continuous updates of the portals. And obviously this can be very useful for further improvements of QuarkOak DB. Regarding the tools, obviously one of the tools that is most important to browse the genome is obviously gene search using specific IDs, and with this a user can retrieve multiple information regarding the gene identifiers, the predicted protein identifiers, and also information about the function of these genes. And obviously it can also, the user can retrieve the information on the complete sequence and coding sequence. In addition, a user can also have a global visualization of the genome, particularly using JBrowse, and in this case, the information about the exon and intron organization can be assessed, and also, if available, other types of alternative splicing forms, so transcript isoforms, different types of gene forms that can occur in the genome. And obviously, also using this JBrowse tool, a user can have access to untranslated regions of the genome, such as promoter regions. Finally, there is the homology search tool using BLAST, so a user can search for closely related as queries that can come from other types or other species, including model species. In addition, to take advantage of the available data sets that were generated for these species, and particularly RNA-seq data sets, we included tools that could be used to assess the expression of these of specific genes under specific tissues or conditions. So to see if a gene of interest could be active or not in a specific tissue or in a specific organ in QuarkOak. So, because together with the EST data that was developed early in 2014, there were other data sets that became available, and they extend in a variety of tissues and organs. So we thought that this could be a good data set to start implementing this tool. We created then, so we pick up this data and align this data to the reference genome in order to estimate gene expression. And in the database in the portal, there is another tool that allows comparative analysis of multiple genes in order to assess the expression of these genes. And the global overview of CorkOak.db, so the front page of the CorkOak.db is shown here. We are continuously trying to improve this portal, and these are some of the things that we are currently doing. One of the things is to link the identifiers of the transcripts that were in the old portal with the genes from the new portal, because we want to keep this information together, because there have been some publications that mention the identifiers from the old portal, and we want to start to make this connection. And also we want to start to integrate curated data from published results, for example, that come from functional validation in wet lab of specific genes. We want to integrate this information in the portal as well in order to improve the knowledge about specific genes. And in the future, we obviously want to prepare the portal to receive other types of data, including genotypic variability, for example, SNPs, and also phenotypic data, among others. And regarding the portal, that's it. So now I give the word to Danielle, that will continue to present other resources from BioData.
So, hello everyone. I am Daniel Faria from Inesc ID. So, unlike my colleagues, I'm not a plant scientist. I'm mostly a computer scientist, but I do have a life science background. So, I'm kind of 2/3 computer scientist, 1/3 life scientist, I would say. And for the past 5 years now, I've been collaborating closely with the plant sciences community, mainly in the activities we've developed in collaboration with other Elixir nodes. So I'm going to talk to you a bit about those activities. Bear in mind that unlike the CorkOak development, these are not 100% national activities. So they are activities in which we collaborated with international partners. They are not only our work, but the work of many people. So before we delve into the actual development of resources for plant FAIR data, let's first dissect the FAIR principles briefly. So what does it mean to have findable, accessible, interoperable, and reusable data? Well, findability is mostly tied to first being able to track objects through their persistent identifiers, making sure that objects are identifiable in a consistent manner across time, that they have enough metadata that you can find them. So a data set should describe, for example, the species which were targeted by the experiment, the type of experiment, the experimental conditions, etc., things that you would need in order to find relevant data sets for you. And obviously, you need to put them in a repository that allows you to search for those metadata. Accessibility means you need to have a specified access protocol that is publicly described, and if necessary, authentication and authorization rules. Interoperability means that your metadata must follow a knowledge representation language that is also well established and libraries or ontologies to describe your metadata and your data. And reusability is again tied to rich metadata, though the level of metadata you need for reusability is much deeper than the level you need for findability. For findability, you only need to find things that are relevant to you, whereas for reusability, you need to have enough metadata to actually understand the experiment. And this should follow existing community standards. And then it also requires that there's a license and provenance information, so you can track the owner of the data and you know what the conditions are for reusing it. So when we started work on the Elixir Accelerate project back in 2015, the state of plant phenotyping data with regard to the FAIR principles was more or less this. So we had a first version of the MIAPPE standard. So this stands for minimum information about the plant phenotyping experiment. It was already providing minimal metadata for findability and for usability. It did not, however, explicitly recommend licenses, and it had the problem that it didn't cover all plant phenotyping domains. So, for example, woody plants were not possible to be modeled under MIAPPE. It did not have ontology recommendations, at least not explicitly. So, whereas there was a crop ontology used by the community to describe the traits and variables being observed in phenotyping experiments, this was not directly linked to MIAPPE. And it was made available in an ISA tools template. So that is a knowledge representation language that is publicly available, but it is not the most user-friendly solution. So it is too, let's say, too advanced for the life scientists, and at the same time not semantically rich enough for the computer scientists. So in a sense, it's not really an ideal solution for knowledge representation. And we had BRAPPE, which is a protocol based on, so a web protocol for data retrieval from databases, established so that the multiple dispersed repositories for plant phenotyping data could exchange data between themselves. And this was recognized as the solution to go forward in terms of allowing a federation of repositories to exist. But BRAPPE was not linked to MIAPPE. So there was clear overlap between the concepts listed in MIAPPE and the calls that BRAPPE allowed you to make, but there was no mapping between them, and there were missing things in both sides of the board. So in a sense, there were several glaring holes that needed to be covered, and several things to improve. And that is what we set out to do in Elixir Accelerate. So the first thing that we set out to do was to improve the MIAPPE standard for several reasons. First and foremost, to make sure that it covered all of the plant use case in Elixir, and that meant a scope extension to englobe woody plants. And that means, for example, features such as identifying biological materials through their GPS coordinates instead of through their germplasm, because when you do experiments on wild trees, you don't know their parenting often, but you do know where they are located, and where they are located won't change. So it's a fixed identifier that while the tree is alive should be persistent. We also revised the structure of the MIAPPE standard to match the ISA data model more clearly. So the previous version did not have an investigation section. So ISA is a data model used by the ISA tools suite. It consists of 3 layers: investigation, study, and assay, that allow you to describe experiments in this nested way. We enriched the examples in the standard, and last but not least, we specified the data model with the figure that you see here on the right, because clearly a standard is more than a flat list of concepts. There are relations between the concepts being listed, and that was an aspect that was missing from the previous MIAPPE version, and it was an aspect that was deterring from us being able to teach how to do it, and to be, if there is no clear way to actually represent the data, or the metadata in this case, it makes it really difficult to have a standard being used. And in addition to these aspects, we also incorporated explicit ontology recommendations. We mapped MIAPPE completely to BRAPPE, and later did the reverse mapping as well. We revised the ISA tab templates and updated them. We also made an OWL encoding to enable plant phenotyping data to be expressed as linked data, so as RDF, and also to enable semantic validation of data sets. And we are also working on a JSON schema version. So in essence, we made sure that we mapped MIAPPE to ISA tools, to the crop ontology, to BRAPPE, and connected it so that it could be exported in RDF. making sure that we are complying with all the FAIR principles. So we are enabling MIAPPE to be expressed through an accessible protocol, that's BRAPI, through knowledge representation languages, both ISA tools and RDF, using controlled vocabularies such as the crop ontology. In addition to this, it was also necessary to develop some ontologies. For example, the crop ontology was missing ontologies for woody plants. It covered traits and features for a number of species, but mainly for crop species. And we were engaged in developing a woody plant crop ontology in collaboration with the Elixir France node. And we also collaborated in developing a rice. In addition to these, and so this is included implicitly in MIAPPE, so all crop ontologies are recommended to describe MIAPPE observed variables. In addition to this, and sort of parallel to our work on MIAPPE, we have been developing the plant experimental assay ontology, which aims to describe experimental procedures and pipelines in plant molecular biology in a semantically richer way. So whereas MIAPPE aims merely to classify and describe data sets, in this PAO ontology, we are actually modeling the whole experimental pipeline. So in addition to working on MIAPPE, we also collaborated in the update of the breeding API, BRAPI, first to make sure that it has full coverage of the mandatory fields, so to make sure that BRAPI is actually a protocol that can be used to access MIAPPE datasets. We also collaborated in BRAPI to ISA tab exporting, making sure that these two forms of representing datasets are interchangeable and compatible, and in validating also the data with ISA tools. And through the PPO ontology, which is an OWL representation of MIAPPE, we are able to export also data from BRAPI to either RDF or JSON link data. In addition to collaborating on the development of the BRAPI specifications, we also set up our own BRAPI endpoint in Elixir Portugal, which is called Pheno. And we did this mainly as a proof of concept, because we had very little data to put in it at first, but this is slowly increasing. So because we had no data to start with, we had to reverse engineer the database from the BRAPI specifications and implement the calls that made sense for the data that we wanted to include there. Right now, we are going to do a shift towards linked data as well. So we are shifting from a traditional SQL database to a triple store in order to facilitate further updates of BRAPI and to facilitate also the interchange of information. So right now, our BRAPI endpoint includes manually created datasets on cork oak, on rice, and on Geotropha quercus. And our BRAPI endpoint was a key component of a federated data lookup service that was set up mainly by Elixir France, but in which we also collaborated for agronomic research. And what this does is essentially import data from all the BRAPI endpoints of Elixir nodes, including Portugal, France, Netherlands, Belgium, and Slovenia, and then display them through a search engine. And it allows also batch download of the data, as well as through web services. So this addresses essentially the aspect of actually findability of data for the purpose of a user that's searching for data sets of other people. By making sure that our data is MIAPPE compliant, we can deposit it in BRAPI compliant databases, because BRAPI is now interchangeable with MIAPPE, and thereby it becomes ingested in FedAir and available for users to. And this addresses the fact that, unlike other domains, plant phenotyping does not have, and probably will never have, centralized databases. So if we don't have centralized databases, the only solution is to have a federation of databases where we enable a central search. So another aspect we tackled, which will also tie into FEDER, is enabling findability of web resources. This is something that search engines such as Google and Yahoo have worked on. They developed something called schema.org, which is used for markup of web pages. And if pages are marked up with schema.org, then Google and Yahoo and other search engines can automatically display some nice information about the content of pages. So this is why when you search for a movie in Google, you see a nice vignette on the right displaying typically the information of the movie and some screenshots of the movie, etc. So bioschemas is essentially an extension of schema.org for the life science domain, and it's something that's a core activity of Elixir. We were not involved in bioschemas at first, so it was mainly developed by people at the Interoperability Platform. But the plant community was asked to participate in a bioschemas implementation study to extend bioschemas to cover our use case. And essentially, that use case would be being able to find pages that describe data sets or pages that actually describe genes, for example, in CorkOakDB, describe genes or describe RNA-seq data sets. In order to be able to find them, we need to mark them up with bioschemas so that search engines can automatically identify the objects that are described in the web pages. And the work that is in progress now is actually to map bioschemas to the MIAPPE fields that we consider that are critical for dataset findability. This regarding phenotypic data. For genotypic data, bioschemas are already well established, but we do need to describe the biological materials also in a MIAPPE compliant way. And we're also going to do a full markup of the CorkOakDB pages with bioschemas once we finalize the set of bioschemas concepts that we want to use for the plant. And last but not least, this will allow FEDER, the Federated Data Discovery Service, to ingest bioschemas markup in order to detect resources that are not BRAPI-based and also absorb their data and allow users to search it. So moving on, we had completed MIAPPE, we published the update. It was considered a major breakthrough for the plant sciences community. And then we realized that it is such a complex metadata standard that people are actually very resistant in adopting it, because despite the name minimal, and this is mostly true of all minimal information standards, they're minimal in the sense that they're the minimum you need to provide in order to have a reproducible and reusable data set. They're not minimal in the sense that they're way beyond the effort that the typical researcher is willing to put into describing their data. And so we realized that people to lower the bar for people to use MIAPPE. We need to make sure that there are user-friendly user interfaces that really facilitate the process of data submission. And we had an ISATAP template, but as I said previously, this was not the most user-friendly solution. So it's a bit too complex for the biologists and not semantically rich enough for the computer scientists. So it's not really catering to either of the two audiences. And so our solution has been, and this is work still in progress, has been to develop MIAPPE templates for popular data management platforms that people are actually using to deposit and register their data, often mid-research. So neither of these platforms, Dataverse or Seek, require you to share your data publicly. You can deposit data there and make it available publicly at a later date. And so we already finalized a MIAPPE template for Dataverse, and it's already available in the Portuguese instance of Dataverse. And mainly the French and Belgian nodes of Elixir are currently working on a MIAPPE template for Seek for Science, which is part of the FAIRDom platform. We are also developing a standalone OWL-based interface for MIAPPE that can be deployed independently of a platform by anyone. In addition to this, again, to try and make sure that people can use this standard, we are providing training on MIAPPE. We already hosted 2 plant data management workshops where we teach participants how to describe a data set using MIAPPE.1. And to do this, we obviously first give some lectures on data management and MIAPPE, but then we have a hands-on group exercise on a simple mock data set using a pre-prepared MIAPPE template that you see here on the right. And we pair people in groups or in groups of 2 or more, which allows people to discuss and exchange their knowledge. And it really makes for an engaging exercise that gets people involved. And we found that this recipe was very successful, so we hosted an event almost a year ago in Oeiras, and then more recently in February of this year in Paris. It was actually the last travel I made this year before the COVID crisis started. We are trying to make a shift towards a virtual event because obviously that's needed now, and we will be planning events for possibly later this year, but for sure early next year. And in addition to these activities, we have also been engaging with industry for several reasons. One of them is that in Portugal, the industry holds a lot of phenotyping data. So for example, the Navigator company with whom we have an ongoing project is a major player in international pulp and paper market, but it also has a research branch that has massive amounts of data on eucalyptus breeding and genetics. So they have genotyping and phenotypic data on over 300,000 specimens, which really dwarfs any publicly available data sets in Portugal. And they were willing to collaborate with us, and they even willing to share publicly some of their data sets. And so the goal of our collaboration with them is to process, annotate, and ingest some pilot data sets into our BRAPI endpoint, and then share with them the knowledge and demonstrate the value of the verification process. For example, by showing them that this allows us to integrate data with other data sets that exist in other BRAPI endpoints. And the idea then is to automate the process so that they can submit data sets in bulk to our BRAPI endpoint if they are willing, and possibly even help them set up their own BRAPI endpoint if they so want it. And finally, we have a plan. This is not yet approved by Elixir, but it's a submitted project, a collaboration with another industry partner, and this is on the technology side. So Phenospec is a company that provides automated plant phenotyping solutions. So they have laser sensors and information systems that allow people to phenotyping data. And as it happens, one of the leading software developers of Phenospec showed up at the training workshop we did in Paris last February, and he was very interested in making sure that their information system is MIAPPE compliant and can export data via BRAPI calls. And for us, this is an ideal partnership. So this is a win-win. This is making sure that data are produced in a fair, compliant way from their origin. So if their automated plant phenotyping platform already produces data in a MIAPPE compliant way and exports it via BRAPI calls, then the end user doesn't really have to do much. They have to, at the start in the Phenospec platform, just describe what they are going to do, and then the platform takes care of everything else. So we are very much looking forward to this collaboration. So putting it all together, our goal has been essentially to have one standard to rule them all. Of course, this is a joke. All these other standards, ISO tools, Dataverse are independent things, but what I mean with this is that we want to make sure that whatever data management tool or data submission platform you are choosing, if there's a template for MIAPPE there, then it should be interchangeable with all the others. So we want to make sure that the platforms that people actually use comply with MIAPPE, so they enable you to submit MIAPPE data, and that all the different solutions are interchangeable. We can exchange MIAPPE data sets in between different solutions. We have also collaborated in defining standards for the genotyping and genomic data, making sure in particular that they comply with the MIAPPE specifications of biological materials to enable data integration between phenotyping and genotyping, which is key, as Celia said earlier, to unlock plant breeding secrets. And also make sure that bioschemas map to upper layers of MIAPPE so that when you have MIAPPE data sets, you can automatically generate markup for the pages describing those data sets. And finally, we are putting all this information together in the Elixir Research Data Management Toolkit, which is being developed as part of the Converge project. This is a project devoted exclusively to data management, where we have and are leading a plant domain use case. Okay, so we want to document all these tools and resources for fair data management of plant data in this toolkit, address the frequently asked questions and the common needs of the community, and prepare data management plant templates for the plant domain that reference these resources that were developed by the community and are listed in the tool. And now I will give the word back to Celia to conclude our presentation.
Okay. So thanks, Daniel. And just before finishing, because we are a bit short on time, I would just like to briefly highlight another resource. It's a portal that is still under active development, although it provides already some functionalities. That is the Plant Small RNA portal. And this portal integrates a pipeline, an automated pipeline, which is the MIAPPE suite, for the analysis of small RNA sequencing data. And these include the analysis of expression and the prediction of small RNA target genes as well. So this pipeline is based on publicly available tools and provides people that are not proficient in bioinformatics a way to automatically and sequentially process their data, generating also reporting output. It also integrates a database that works as a repository of small RNAs that are annotated either by the MIAPPE suite pipeline or other small RNAs that are publicly available. And the development that of visualization of data related to microRNA target gene interactions in order to improve functional predictions. So, but as I said, this is still under development. And to finish, I would like to say and highlight that all these efforts that we have been describing will only be effective if they are indeed adopted by the potential users of the plant sciences community, both in the academia and industry. And as more and more researchers adopt these resources, the easier it will be in the future to find data, to access data, and to reuse that data. So we know this is not a trivial task. people are resistant to the use of these standards because it involves more work, of course, but that is why we are trying to make it as easy and painful and, and painless, not painful, as possible. So in order for this to happen, we are investing a lot on making available training materials and organizing training sessions so that potential users of these resources are acquainted with the resources and to implement best practices in plant data management. Of course, this is a key activity, the training, but also we are engaged with other more focused communities. For instance, the emphasis, which is the European infrastructure for plant phenotyping. And we have, the Elixir has a collaboration agreement with emphasis for these ends, but also with other communities like the crop ontology, the BRAPI, and the biodiversity communities. All these activities are aligned with the work going on in other Elixir nodes, with which we collaborate through not only European projects such as Converge or previously the Accelerate project, but there are also other instruments that facilitate these interactions by providing funding, which are the implementation studies and knowledge exchange programs with the industry and also staff exchange programs. So just to finish, I would like to acknowledge all the people that have been involved in these activities from BioData, Plant Sciences Community. So a lot of people here have been involved from faculty, from IBET, from the ITKB, IGC, and also past collaborators like Daniel Sobral and Sirenia, but also very special thanks to our partners in other Elixir nodes, Cyril Pommier and Françoise and Celia Nicotet from Elixir France, also Richard Finkers and Evangelia from Elixir Netherlands, and Frederick Coppens and Paul Kersey from Belgium and UK respectively. So thanks for your attention, and we'll be happy to answer any questions that you may have.
Thank you, Celia. Thank you, Danielle, and thank you, Pedro. So as Celia said, I would like to ask the audience if there are any questions related to this topic. So please raise your hand or unmute yourself to ask the questions, please. You can put it on the chat as well. Okay, so if there are...
Yeah, of course, people can contact us some other time by email or through BioData channels if they are interested.
Exactly. Okay, so if there are no questions, I would pass the word to Anna again. And I will share my screen.
Okay. Thank you all for coming. I take the opportunity to announce the next BioData talk that will be on the 17th of November at the same time. And thanks to the speakers and thanks to the audience, and have a nice evening.